Hi, it's Dr. DeBusk, and in this video I'm going to talk about the parts of the cell and mainly the parts of the plant cell. First, I need to talk about the different types of cells. Cells can be classified as either prokaryotic or eukaryotic cells. Prokaryotic cells are simpler and often smaller than eukaryotic ones and are found in the domains of bacteria and archaea. Eukaryotic cells are found in plants, animals, fungi, and protists. Both cells share some similarities in that they both have cell membranes, cytoplasm, and in the case of plants and algae, cell walls. They also both have ribosomes, which are used to synthesize proteins. They both have DNA, but prokaryotes have circular DNA that is coiled in a nucleoid, while eukaryotes' DNA is in a membrane-bound nucleus. Eukaryotic cells have many organelles that allow them to be more diverse and complex. In this concept map of the plant cell's organelles, I group them into four broad categories of function. First, the cell manufactures substances. We have the nucleus, which is the control center. In other words, it has the information, the blueprints, and it's gene controlled. It's telling other parts of the cell what to do. If we work outward from that, we have the ER, the endoplasmic reticulum. We have both the smooth ER and the rough ER. Now the rough ER is rough because it has little ribosomes on it. So what do the ribosomes do? Those are going to be where we produce proteins. So it's the site of protein synthesis. The rough ER gives us a lattice where we can actually do that, and we can make different constituents of the cells. These parts and chemicals are going to be used throughout the cell. Before we continue with rough ER, the smooth ER is responsible for making lipids. Almost all parts of the cell are going to be made up of lipids, since all the membranes, including the cell membrane and all the membranes around the organelles, are made up of phospholipids. So the smooth ER is super important. Some of the ribosomes float out in the cell, but most are going to be found right on the rough ER, and they are going to make proteins. Now those proteins will be trapped in transport vesicles and brought to the dictyosomes. From there, they can go to different parts of the cell or outside of the cell. To break down substances, one of the main organelles is the central vacuole. It is used for storage of materials. It also allows plants to regulate their amount of water. Microbodies also break down substances. Peroxisomes detoxify peroxide into water and oxygen, and glyoxysomes convert stored fats into sugars. So let's get to each of those. We'll get to the other organelles in part 2. The nucleus can make up to 50% of the cell in dividing cells, like in these root tip cells. It stores the organism's genetic information and is surrounded by the nuclear envelope. They contain a nucleolus where components of ribosomes are synthesized. The nucleus carries out information retrieval by making copies of specific parts of the DNA by making messenger or mRNA, which moves out of the nucleus through the nuclear pores. The mRNA then moves to the ribosome. The ribosome is made up of 2 proteins and tRNA. What ribosomes do is translate the message of the messenger RNA to produce protein. This can take place in the cytoplasm or on the endoplasmic reticulum. The endoplasmic reticulum is usually attached to the outside of the nucleus. It's a membrane that folds upon itself. It's going to have ribosomes that sit right on that so it can synthesize proteins. Those proteins end up going to the inside. So those little green dots here would be showing proteins or bits of proteins that are put together. Most plant cells don't have many ribosomes, but some cells produce a lot of ribosomes, such as protein-rich cells of legumes, including peas and beans, and cells that produce protein-rich mucilage, like this hair cell. This is what rough ER looks like underneath the microscope. There's also smooth ER, which doesn't have ribosomes on the outside. Mostly it's where we synthesize lipids inside the cell, but the function of the ER is to give us a lattice so we can build things. If we're talking about a cell as a factory, the ER is the factory floor with the workers fitting on top of it. Next we go to the dictyosomes, also called Golgi apparatus. They are a stack of thin vesicles. In general, it takes information that's produced in the endoplasmic reticulum and it shifts those to the different parts of the cell. So I like to think of it as the UPS of your little factory. Materials made in the ER, repackaged, and things added like in cellular UPS. Then it moves to different parts of the cell or even outside of the cell. A mucilage cell, like this one shown here, has a lot of dictyosomes. The fusion of the vesicles with the plasma membrane is so abundant that the plasma membrane appears scalloped. You can see the mucilage stained purple outside of this root cell. Moving to the central vacuole, it is often the largest part of a mature cell. It consists of a single membrane, or tonoplast, and it stores water, salts, crystals, starch, protein bodies, and other granules. It is critical for cell enlargement compared to animal cells, which are required to build another cell to grow. For example, as flower buds open, petals expand from tiny to full-sized in just a few hours by simply enlarging their vacuoles. The pigments of these flowers can also be in the vacuole. A system to excrete waste never evolved in plants, so metabolic wastes are stored in the central vacuole. So it essentially goes to the bathroom inside of itself. Some of these wastes are toxic to animals, such as graphite crystals and tannins, tasting bitter and keeping animals from eating them. Continuing with the breakdown of substances, peroxisomes are involved in detoxifying certain byproducts of photosynthesis and are found closely associated with chloroplasts. Glyoxysomes are found only in the oil-rich seeds, and they contain enzymes that break down the oils to be used as energy during germination. These microbodies are often composed of a membrane, matrix, and crystalline core. This was the first half of the organelles. I hope this was helpful.